Hi everyone, it's a pleasure to be with you here today. Thank you for joining us. So I will be presenting a tiny bit what we were doing during 2022 for our Tundra Roadshow. But first of all, I want to highlight again the beauty of Cryo-EM. Probably everyone here knows how important is the visualization of the building blocks of life functions. So, the human body contains between 80,000 and 400,000 proteins. And those proteins are very important. They are essential for life functions, from growth, repair, metabolism, structure, and to disease resistance. So understanding the structure of those proteins is very, very key to be able to understand not only the diseases, but to develop new therapeutics to treat those diseases. So obviously, Cryo-EM, as you know already here, is the fastest growing structural biology method for characterizing proteins and pathogens. And I would like to show you, how, or some of those examples of how Cryo-EM is impacting research in human health. So the first example I want to show you today, you can see here in the left side, was working in collaboration with the MRC lab in the UK, and using Cryo-EM single particle analysis, we were able to understand and discover a new tautopathy. So it's a neurodegenerative disease. And we were able to, doing this, isolating from brain patients the filaments and analyzing the Cryo-EM structure of those filaments. So this was a very important discovery using Cryo-EM single particle analysis. In the middle, I have another example for you. This is better therapies against Ebola, and this work was done in collaboration with La Jolla Institute in San Diego, in California. So we were able, doing this, again, Cryo-EM single particle analysis, to really understand and to really discover two human antibodies, like we're able to target these Ebola viruses. Like they are very, very lethal, as you already know. And the latest example I want to show you today is how, using Cryo-EM single particle analysis, we can really understand cancer and we can really discover new therapies. In this particular example, I want to focus on the kinase. This Janus kinase is a membrane protein. Like, it's very, very challenging to do and to get the structural information by crystallography. As you know, membrane proteins can be very tricky. So using Cryo-EM single particle analysis, we were totally able to really understand the structure of this protein and to really develop new therapeutics and new therapies. So now, like I show you, and I hope you understand the importance and the impact like Cryo-EM is playing, nowadays in the structural biology arena, as well as the scientific community. I would like to show you right now as well, how Tundra, specifically, or entry-level single particle analysis microscope, is playing as well a very important role in this structural insight or structural biology arena. So Tundra is, as I say, a site entry level, and it's an entry-level single particle analysis microscope that was launched in November 2020, and it's very easy of use and has artificial intelligence. So even for people like they are no experts in electron microscopy, they can definitely and very easily use our Tundra. So I can show you here some of the examples of the structural biology information you will be able to obtain with the Tundra. For example, we have in this case a 330 kilodalton membrane protein, like we were able to obtain using the Tundra at 4.2 angstrom resolution. You can see as well in the middle the 200 kilodalton membrane protein GABA receptor, like it's a very important target, for developing new therapeutics against cancer. And we were able to obtain this 3D volume at 3.4 angstroms. Again, 700 kilodalton proteasome, another very important target for different therapeutic, developments. And using Tundra, we were able to obtain a 3 angstrom resolution of this protein. And you can see other examples here at the bottom. For example, the ribosome at 3.5, the AAV at 3.5 as well, and of course, apoferritin like is the test control sample for single particle analysis at 2.6 in only 6 hours of data collection. So this is a very impressive, instrument where you will be able to screen, optimize your samples very easily, very ease of use, and you will be able to obtain already very important structural information. With that being said, I want to tell you as well, like in 2022, we were able to schedule different Tundra roadshow maps, and you can see like Tundra is coming to your institution or maybe have been already there. So those are the places like we schedule Tundra workshops, Tundra presentations during 2022. But I want to reassure you, if you miss some of these workshops, you don't need to worry. We are planning to keep moving with those Tundra presentations and Tundra virtual demos in 2023. So please, I am hoping like you will be able to join us in 2023. And with that, thank you so much for your attention today.
Thank you very much to the Nanoport for that fantastic demonstration. And here, this will just be a summary slide before we transition into some question and answers. But really what I'm trying to highlight with this slide is just the broad range of applications that the Tundra can assist with, and cryo-electron microscopy more generally is able to help with. And so obviously you can do so-called traditional structural determination using cryo-electron microscopy in the Tundra specifically. And we have some examples here that Natalia, during her introduction, elaborated on in more detail. But there's one area that we didn't touch upon, but cryo-electron microscopy can really help with nanoparticle characterization. And so we have some examples here of how that's able to aid research. And we can answer some questions about that in the Q&A if there's any questions there. And really the key point that you would have seen during the demo is just how straightforward and accessible the Tundra is. And so we have a diagram here in the sample optimization line where you're using that structural information that you're gaining from the Tundra to help optimize your sample. And as you saw with the nice resolutions that you can get on the Tundra, you can obviously be satisfied in some cases with that level of data. But if you require to move to a higher resolution, well, you have already optimized your sample. So when you go to a Glacios or a Krios, some other instrument, you then have a lot more confidence that your sample is going to be able to achieve whatever resolution you're looking for. And within all of this, when you saw the Nanoport operating the Tundra, I think probably the key takeaway was just how straightforward and accessible the microscope is. So it's possible that some people approach Cryo-EM with the idea that it's going to be a little bit difficult and maybe not so accessible to a range of different researchers and labs, but really you can see just how straightforward and how much effort has gone into really optimizing and improving the process so that anyone more or less can access cryo-electron microscopy and operate these microscopes. And so I think with that, just once again, thank you to the Nanoport for the great demo, and we'll transition into question and answers. Thank you.
Hello, everyone, and welcome to our 2022 Labroots webinar series. My name is Tara Niles, and I am the Regional Market Development Manager here for the Thermo Fisher Life Sciences Electron Microscopy team. We're so glad to have everybody here with us today. During today's session, we have an exciting lineup prepared for you today. It's a combination of presentations from our scientists, as well as a demo from our Nanoport of the Tundra, the Thermo Fisher Scientific Tundra Cryo-Transmission Electron Microscope. So for today's webinar, we encourage you to ask questions. We'll have some time for Q&A at the end, so please do feel free to type any questions that you might have in the question box and click send. If for any questions that we don't have time for today, we will follow up with you via email to make sure that all of today's questions get answered. We've had a really busy 3 months since the Tundra instrument has been installed in the Nanoport. And over the last 3 months, we've had many events at universities around the US. So these events have included speakers doing scientific presentations, as well as the Nanoport connecting to do a live demonstration for all of the attendees. The sessions that we've had have been extremely well received, and we've had a great number of users, anywhere from starting users to Cryo-TEM, all the way through to directors of Cryo-EM facilities looking to expand their capabilities using the Tundra Cryo-TEM. Now I'd like to introduce our speakers for today. Natalia is a biochemist and protein engineering specialist with more than 15 years experience in single particle cryo-electron microscopy, as well as immunogenic design efforts for vaccine achievement. She received her doctoral degree in biochemistry from the University of Leuven, Belgium. In October 2017, she joined NCI as the director of the EM lab and the team lead for Cryo-EM section at the Center for Molecular Microscopy. She was responsible for the management of a wide portfolio of projects related mainly to cancer and HIV diseases. She has authored over 70 publications in high-impact scientific journals, such as Nature and Science. She joined Thermo Scientific in January of 2021 as senior scientist, senior product specialist for electron microscopy. In her current position, she is supporting EM in the East Coast and Midwest US. Doing today's demo is Veer Bhatt, who is an application scientist in life sciences at Thermo Scientific. He obtained his MS in biophysics from All India Institute of Medical Sciences, New Delhi, and his PhD in biophysics from The Ohio State University. As an application scientist, he conducts training, performs data acquisition, and delivers demonstrations on various TEM workflow-related products in the life sciences. Prior to joining Thermo Scientific, Veer utilized his expertise in structural biology, biochemistry, and biophysics to contribute to cancer research. His work led to understanding how cancer-causing virus hijacks human protein machinery to leverage integration of viral DNA into human DNA. His work on intracellular membrane remodeling and carcinogenesis revealed novel details of how BAR family of proteins deforms membranes in various processes such as autophagy and apoptosis. And our final speaker for today is Zuben Brown, who obtained his undergraduate degree from the University of Sydney, Australia, and his PhD in structural biology from Osaka University in Japan. His postdoctoral work was completed at Columbia University in the lab of Nobel Prize winner Joachim Frank, and he uses Cryo-EM to study the structure of hepatitis C virus internal ribosome entry site as it hijacks the canonical cellular translation machinery. He has experience with all aspects of Cryo-EM project, including operation of high-end microscopes, data collection strategies, computational analysis, and model building. He recently joined Thermo Scientific as senior product specialist. In today's session, we'll start off with a brief introduction to Cryo-EM, followed by a live demonstration from our Nanoport, and then wrap up as we move into our Q&A session. Now we're ready to learn more from our speakers. Natalia, over to you to start us off.
Hi everyone, it's a pleasure to be with you here today. Thank you for joining us. So I will be presenting a tiny bit what we were doing during 2022 for our Tundra Roadshow. But first of all, I want to highlight again the beauty of Cryo-EM. Probably everyone here knows how important is the visualization of the building blocks of life functions. So, you know, the human body contains between 80,000 and 400,000 proteins. And those proteins are very important. They are essential for life functions, from growth, repair, metabolism, structure, and to disease resistance. So understanding the structure of those proteins is very, very key to be able to understand not only the diseases, but to develop new therapeutics to treat those diseases. So obviously, Cryo-EM, as you know already here, is the fastest growing structural biology method for characterizing proteins and pathogens. And I would like to show you, how, or some of those examples of how Cryo-EM is impacting research in human health. So the first example I want to show you today, you can see here in the left side, was working in collaboration with the MRC lab in the UK, and using Cryo-EM single particle analysis, we were able to understand and discover a new tautopathy. So it's a neurodegenerative disease. And we were able to, doing this, isolating from brain patients the filaments and analyzing the Cryo-EM structure of those filaments. So this was a very important discovery using Cryo-EM single particle analysis. In the middle, I have another example for you. This is better therapies against Ebola, and this work was done in collaboration with La Jolla Institute in San Diego, in California. So we were able, doing this, again, Cryo-EM single particle analysis, to really understand and to really discover two human antibodies, like we're able to target these Ebola viruses. Like they are very, very lethal, as you already know. And the latest example I want to show you today is how, using Cryo-EM single particle analysis, we can really understand cancer and we can really discover new therapies. In this particular example, I want to focus on the kinase. This Janus kinase is a membrane protein. Like, it's very, very challenging to do and to get the structural information by crystallography. As you know, membrane proteins can be very tricky. So using Cryo-EM single particle analysis, we were totally able to really understand the structure of this protein and to really develop new therapeutics and new therapies. So now, like I showed you, and I hope you understand the importance and the impact like Cryo-EM is playing, nowadays in the structural biology arena, as well as the scientific community. I would like to show you right now as well, how Tundra, specifically, or entry-level single particle analysis microscope, is playing as well a very important role in this structural insight or structural biology arena. So Tundra is, as I say, entry level, and it's an entry level single particle analysis microscope that was launched in November 2020, and is very easy of use and has artificial intelligence. So even for people like they are no experts in electron microscopy, they can definitely and very easily use our Tundra. So I can show you here some of the examples of the structural biology information you will be able to obtain with the Tundra. For example, we have in this case a 330 kilodalton membrane protein, like we were able to obtain using the Tundra at 4.2 angstrom resolution. You can see as well in the middle the 200 kilodalton membrane protein GABA receptor, like it's a very important target, for developing new therapeutics against cancer. And we were able to obtain this 3D volume at 3.4 angstroms. Again, 700 kilodalton proteasome, another very important target for different therapeutic, developments. And using Tundra, we were able to obtain a 3 angstrom resolution of this protein. And you can see other examples here at the bottom. For example, the ribosome at 3.5, the AAV at 3.5 as well, and of course, apoferritin, like is the test control sample for single particle analysis at 2.6 in only 6 hours of data collection. So this is a very impressive, instrument where you will be able to screen, optimize your samples very easily, very ease of use, and you will be able to obtain already very important structural information. With that being said, I want to tell you as well, like in 2022, we were able to schedule different Tundra roadshow maps. And you can see like Tundra is coming to your institution or maybe have been already there. So those are the places like we schedule Tundra workshops, Tundra presentations during 2022. But I want to reassure you, if you miss some of these workshops, you don't need to worry. We are planning to keep moving with those Tundra presentations and Tundra virtual demos in 2023. So please, I am hoping like you will be able to join us in 2023. And with that, thank you so much for your attention today.
Welcome to the Tundra demonstration. I'm Veer Bhatt. I'm an application scientist with Thermo, and I'll walk you through the demo. Let's get started. So there are three elements to our system: the microscope, the cryo-loading workstation, and the microscope PC. We will start with cryo-loading workstation. And to begin with, we have our on-system display, which is right here. I'm going to share a live copy of it for sake of clarity. So let's move to share my screen. So here there are three buttons on the left pane, starting with status. We can see that loading workstation, transfer device, and the microscope all are good. We can see some additional details. For example, the cassette has a sample in slot number 4. The microscope is already at liquid nitrogen temperature, and we have an estimated time of 22 hours remaining on it before we refill it with liquid nitrogen. Then the second button is actions, which hosts most of the actions that will need to be performed. Load, unload samples from the cassette, set temperature mode, load sample to the microscope, unload sample, refill liquid nitrogen, cryocycle loading workstation. Maintenance is of course reserved for our system engineers. And then there is an available recovery button should there be any misalignment in the loading workstation. You will have an option to try to recover it by pushing the recovery button. The cassette gives details of what samples are in the cassette. One of them will be loaded into the microscope. So in addition to all these details, I also want to emphasize that there are several animations that come along with every single action. For example, if I need to refill liquid nitrogen, let me push this button. A guided animation tells me that I need to open the valve on the nitrogen tank counterclockwise, and then there are 4 sensors in the loading chamber which are updated live in this animation, and the 4 sensors correspond to 4 levels. We need to have liquid nitrogen up to level 3 for us to be able to manipulate the sample and transfer our grids to the microscope. So just like this, every action has corresponding animations which are very helpful for new users. So with that, if I'm to start out an experiment, the first thing I would do is to set the temperature mode. So I'll push this button and select either negative stain or liquid nitrogen temperature. For today, I have already selected liquid nitrogen temperature because I am working with cryogenic samples. So I'll proceed with that, And then let's move to see more details of the procedure of loading the grid to the microscope. So here, the next action to be performed is on system display, where I say load, unload samples from cassette. I'll push this button and the cassette will move out. There are several cutouts in this loading chamber. There are 5 cutouts for your autogrid boxes, so to speak. Here there is a dedicated cutout for your cassette. The cassette is identical to that used in Glacios and Cryos, but here it is an integral part of the loading workstation, so it does not come off. And another point to remember is that only slots 1 through 5 are available for the transfer device. So your grids that you load into slots 1 through 5 can be used to transfer to the microscope. There is a dedicated cutout for clipping as well. So if you want to sandwich your grids in place, you can do that here. And then there are some cutouts for your tools to be stored while you're using it. So here, let me transfer a grid. So this is indeed an autogrid, and it is fully compatible with Glacios and Cryos as well. I'm loading it into slot number 3. Let me check if it is properly engaged. And once that is done, I will again go back to my on-system display. I loaded the grid in slot number 3. I can edit some details if I want to here, and if I need to, I can edit some details and then continue, and the cassette will return to be ready for the transfer device. So here then, the next action is, of course, I want to load a grid to the microscope. Again, I push this button that says load sample to TEM, and then I select one of the 5 slots. There are only 2 grids here in slots 3 and 4. I select slot number 3 and I say transfer sample to TEM. And then a countdown starts, which takes about 8 minutes to complete. During this countdown, the first thing that will happen is that the loading workstation tells the microscope that a grid is coming to you, so be ready. In order for microscope to be able to receive the grid, the turbo pump on the microscope starts ramping up to minimize the airlock pump time. Then again, what's happening on this end is the transfer device, the semi-automated loader housed in the transfer device, is being physically cooled by its own thermal mass for first few minutes, and towards the end of the countdown, the gripper arm in the transfer device will grab the grip and return to the housing in the transfer device. That will be followed by an evacuation for about a minute. So your grid will be kept contamination free in the transfer device. As I just said, cryo-loading workstation talks in the background to the microscope all the time. So they are in synchrony. While the countdown is being completed, let's go through some additional elements of both cryo-loading workstation as well as the microscope. So to start with, of course, there is the on-system display, which we described in detail just a while ago. Then there are 2 places on either side of the transfer device where you can store your tool. Just as you can see here, the slots are heated, so both of the pads are heated, so your tools are ready to be used whenever you are. In addition to the storage area for tools, we have heated pads here below the bench top as well, so that you can work comfortably. The level of cryo-loading workstation can also be adjusted. There is a lever provided here to do that if you're not comfortable with the height setting. There is an emergency shut-off button available, and what you cannot see on the screen is a reset button available here. Indeed, the most important part of the cryo-loading workstation is right here, the semi-automated loader, which is housed inside the transfer device, and which we'll see in action in just a little bit. With that in mind, let's move on and take a walk around the microscope. So I'll start with the front. The microscope is fully enclosed, and there is a dedicated on-system display available on the microscope. It has 4 buttons. The most useful button available on on-system display is the cryocycle button. So here, cryocycling the microscope becomes super easy. You can just push the button and the cryocycle is completed. You do not have to worry about emptying the microscope viewer or drying it, unlike some other systems. There is an emergency shut-off button available right here, and this is the housing, so to speak, where the tunnel of the containing the semi-automated loader is connected on the microscope, and the transfer device will enter. Right. With that, let's take a look at the back side of the microscope and some other details. So on top, we have the electron source, which is a 100 kV XFEG in this case, followed by the accelerator stack and the column here. And this is where the beam interacts with your sample. It's your objective lens system with the electronically controlled apertures right here. Following that is your projector lens system, and towards the end of the column, first the flu screen, and then your CETA-F camera. CETA-F camera writes 14 frames per second and enables you to get a 4 times enhanced signal-to-noise ratio relative to the CETA-16M camera, for example. The vacuum system is oil-free in the system. So we have, for example, ion getter pumps for the gun and column. We have turbo pumps, both for airlock as well as cryocycling. As I said just a while ago, the microscope viewer is an integral part of the microscope. It can be cryocycled with push of a button. It has a capacity of 5.5 liters, and one complete fill of the viewer lasts about 3 days, and that enables you to acquire SPA data easily, which is something that is quite difficult for your traditional side entry systems. With that, we can return to our countdown. Towards the end of the countdown, the grid returns to the semi-automated loader, and the transfer device is evacuated. So your grid is kept contamination free. So here the actions I'll need to perform are simply to take the transfer device out and insert it in the tunnel on the microscope. So here the lights on the microscope are blinking. Just a simple visual cue for me. and the transfer device is inserted. So during this process, the grid will be transferred from the transfer device to the stage. And as you can see, this process is quite a lot simpler than our traditional side entry cryo holders. Another thing that some of you may already appreciate is that compared to side entry holders, it's relatively easy to insert and as well as if you want to recover a grid. It is also quite easy to perform a long SPA data collection, and that is because traditional side entry cryo holders, they do require constant refilling of liquid nitrogen, whereas here we once the transfer is complete, we return the transfer device back to its housing in the cryo-loading workstation, and the grid is protected and kept safe in the microscope itself. So here, once we return the transfer device to the cryo-loading workstation, there it would be returned to atmospheric pressure and vented with dry nitrogen to keep it contamination free and ready for next transfer from the microscope. This process takes about 2 minutes. So in addition to the 8 minutes of the initial countdown, the whole process takes about 10 minutes for a complete transfer. So the actions here are again quite simple. I take the transfer device out, blinking lights, a visual cue, and I insert it in the loading workstation. It's as simple as that. So with that, we can move on to see how data is acquired or how screening is performed using Tundra. So I'll share my screen for that. So right off the bat, we see that there is a simplified version of APU available here for Tundra. We have available two things that I already want to point out. One is the choice of experiment type. We have screening, medium resolution, and high resolution. So if we look at these experiment types, in screening we have a larger field of view, smaller pixel size, so that enables quick screening of our grid for ice quality and such. For high resolution, in contrast, we have a field of view that's about 3 times smaller, and also a smaller pixel size that increases your chances of getting high resolution structures. So in addition to that, what simplifies the workflow of Tundra significantly is availability of traffic lights. So here, most of the components of the microscope are already covered. So for example, vacuum, source, acquisition, column optics, storage, loader, column, and alignments. And how are alignments performed? It's as simple as pushing this recover button. So if there's anything off, the recover button becomes available and the microscope can recover itself. Specifically for alignments, here, if I look, the traffic light has already turned blue and some of the alignments are expired. So in this scenario, if I push the recover button, all the alignments will be recovered. And this already illustrates the contrast of Tundra with several other systems where you need significant training and some expertise to perform all these alignments. So here, let's start with acquiring an atlas first. We click a new session and give atlas a sensible name. And click start. Atlas acquisition takes about 3 minutes to complete. During that time, let's take a short tour of EPU. So we start with the landing page. As I just described, it houses 3 different experiment types, and if we want, we can modify them. So here, for example, I have modified some of them, and it's as simple as making any modification and clicking on the plus button, and then I can save my modifications as I wish. And the next important feature, as I discussed in detail, is the traffic lights button, which makes recovery as well as alignments very streamlined. So it's all done with push of a button. Then the SPA workflow in general goes from left to right and top to bottom. Here in the taskbar, there are only 4 available tabs: Preparation, Atlas, Auto Functions, and EPU. For a typical Tundra workflow, you really need to go to only Auto Functions and EPU tabs. Auto Functions, if you, after screening, you like your grid and you want to acquire a full SPA data set, you would go to Optimize Beam for some finer alignments, such as astigmatism, coma, finding parallel illumination, etc. And all of those alignments, again, will be performed with push of a button. So we say Optimize Beam and push this Start button. We can select an aperture, objective aperture, if we desire to do so, and then apertures will also be centered as part of the alignment. And the last tab is EPU, tab of EPU, which is where we set up the automated data acquisition. Let's look at some more details and an overview of what goes on here. So each tab has a dedicated ribbon to it, which has a group of functions. For example, for preparation, we have all these presets that here are predefined for you because we did select an experiment type. So all of the presets, for example, Atlas preset, they have dedicated optics settings like exposure time, magnification, illumination mode, there's microprobe or nanoprobe, size of the aperture, etc. that is associated with it. If we move down here in the taskbar, available to us are calibrate image shifts and Atlas optics alignments as well, a rarely used option. Moving to the right, we have Atlas tab, which is acquiring Atlas right now. And again, this is the imaging area where all images appear through Atlas acquisition as well as your EPU automated data acquisition run. Then are a few boxes on your right side. To start with, there's a box for notifications here on top. There's the status box, which is a live log of what the software is doing at the moment. There is a histogram that is associated with each image, and there is the image metadata here. So here, Atlas acquisition is already complete, and following the completion of Atlas acquisition, there is a clustering that EPU executes to group grid squares that have similar ice quality. So here, for example, I'm thinking that the red grid squares, they have too many broken areas, so I don't like red. From what are left, let's say that I like cyan grid squares. I know the sample does like to go to thicker ice, so I leave cyan selected and I unselect all the other colors. And with that, I can move on to set up an EPU data acquisition. So I create a new session. I'm asked if I want to use the preferences that are saved. I would say yes, that's a time-saving feature. I give the session a sensible name. I would, between automated and manual, I select manual. That will allow me some additional customization of selecting holes via a selection brush. Acquisition mode, accurate versus faster. I'll select faster, so I can use aberration-free image shift that moves the beam to the neighboring areas of the sample, as opposed to moving the stage, increasing the throughput of the data acquisition significantly. I also have the option to live monitor the data that are being acquired. To do that, I need to connect the EPU session to EPU quality monitor, which is housed in Athena. So right now I am connected to Athena, and I can, if I want to connect to a particular location in the EPU quality monitor, and then the data that are being acquired will be live processed, and I'll have a better insight on different metrics, for example, CTF, astigmatism, etc., that my images do have. For this demo, we are not doing that. So here I give a location to the data that will be written to the offload server. We say apply. The taskbar is populated with new options here. So I go to square selections, and here you can see that the cyan color that I selected is already correlated to the EPU session. I can further deselect some grid squares if I want, or I can unselect all of them and select a few grid squares if I want to. The next function is pole selection. So here I just go to auto eccentric. That will drive the stage automatically to a selected grid square, as we can see here in the status box. And once the stage move is complete, eccentric height will be found automatically. And that procedure invokes applying positive and negative beam tilts and maximizing the correlation between the images coming from positive beam tilt and negative beam tilt. The point where the maximum is reached is the eccentric height. Once the eccentric height is estimated, it will be populated here at stage Z, and EPU will remember the eccentric height for that grid square. For the remainder of the data acquisition. Once the eccentric height is found, a new image will be acquired and will appear here. And as I said, this is a live log. I like reading what's going on here at the current moment. And here already we see that the ice quality is reasonable enough. It is on the thicker side, but that is necessary for this sample. And we can go to the next button here, measure hole size. I can increase the contrast of the image to my liking, and let's say find holes. And if there are a couple of options here, I can tweak the holes that are selected by play at the histogram. So I can move the sliders from left or right to select more or few holes. So the slider on the right to remove dry holes if there are any, and the slider from the left to remove the holes that have thicker eyes. I can also perform the selection right from the screen, where I can set the ice filter lower bound or upper bound. And I can also not select all the holes, but select some of the holes within the window of grayscale that I have selected. How do I do that? I use this fast screening option here, turn it on, and select a number of locations. So here, for example, I can tell EPU that collect data from a maximum number of 10 locations. And that is an approach that I can take for screening purposes. So here, let me turn that off. Let's move stage to an area where holes are selected and move to the next step, which is defining the template. Push the acquire button, and that will acquire an image at eccentric height optics setting. And here there are two areas that already appear. One is where my image, my data would be acquired, and second is autofocus area. Both of these are populated from my saved preferences. So you can see that the list of defocus values is already populated here for the data acquisition area, and the routine that I would like to use for autofocus is also already populated here. So for example, here I'm employing autofocus after a distance of 10 microns. Another option is to do it every single time, to do it never, or to do it once per office cluster. If I want, I can of course tweak my template, and let's, for example, say after centering means autofocus will be performed once per office cluster. And let's try out this template. So if you will start by taking an image at whole eccentric height preset and centering the area of acquisition automatically, following which autofocus will be performed. Again, beam tilts will be employed, positive beam tilt and negative beam tilt, and cross correlation between the two images will be used to estimate focus. Right, and there's our image. And here already I can see that some of the particles do look quite good. So this is side view of KLH, and this is view from top down. KLH does have D5 symmetry, which means C5 propagating along an axis. So this is the view from top down, and this is the view from side. In addition, what I can learn from this already is that there are a lot of disintegrated particles. So several of the subunits, in fact, many of the subunits have already disintegrated. And what that tells me that I do need to revisit biochemistry. So for example, here I can perform an additional sizing step before I collect a full SPA-driven data set on the sample. At this moment, in contrast to revisiting my biochemistry, if I were already happy with the sample, I would perform some additional alignments before acquiring a full SPA data set. And how would I do that? I would go to Auto Functions tab. I would select an aperture, let's say 100 micron aperture, and say set, and then click on the start button. So this is something that I again want to emphasize, that even in the scenarios where you want to collect a full SPA data set, all the alignments are fully automated and performed with a push button. So here the program will start with finding eccentric height for the sample for every single grid square, and then perform all the other alignments, such as minimizing astigmatism, autocoma, finding parallel illumination, centering the apertures. Should you use an objective aperture, that will be centered as well, and all that will be performed fully automatically. So there are two levels of screening here. The first one was Atlas acquisition, and it takes from first inserting the grid to the microscope, into the microscope, getting to the point that an atlas is acquired. It takes about 10 to 15 minutes, and this already gives us insights about ice quality and whether we want to proceed with the grid. So here, for example, at the level of atlas, the ice thickness does look promising enough for us to proceed to maybe either screen further or maybe even acquire some data. And here, when we took an image at higher magnification, so to speak, we noticed that the particles are disintegrated somewhat. The ice quality is good. So what that tells me, for example, is that my vitrification conditions are good, but my biochemistry is not. So I need to revisit biochemistry before I can get best possible resolution from this data. And that's the information as an example that I could get from this sample. So with that in mind, I also want to introduce another software that that is available to you for Tundra. Indeed, everything in the workflow, as I just showed, was performed out of EPU, whether it were the alignments or data acquisition setup. But there is another software called Microscope Companion, or MICO for short, which is available, and it does house some nifty little functions, and a couple of them are important and fundamental to your workflow. So what is available here is, for example, your column overview, vacuum status. So you can, of course, close your column valves from here. You can execute cryocycle from here. Status of electron source, high tension, status of stage, alignments. This is something that becomes quite interesting should you want to live monitor what's happening while EPU is executing alignments. So there are two levels of alignments, as I said, from EPU. When you push the recover button in EPU, EPU performs a set of alignments which we call daily alignments. Should you want to acquire a full data set, EPU performs a set of alignments called optimized beam alignments, and they can all of these can be live monitored in MICO. And this is where this is the only place where MICO becomes fundamental or required to be used, which is to acquire reference images. So here it's again done with push of a button and takes only a couple of minutes. I need to measure dose, and once I am in the correct dose range, I need to acquire a full set of gain reference. And the last button is an optics button where you can measure beam diameter. To conclude, in Tundra, we see that it's very easy to use, even if you are not an expert or if you're not familiar with electron microscopy in general. It's quite easy to transition to electron microscopy, and also in Tundra it becomes quite easy to not just screen your grids, but also to collect big single particle analysis driven data sets should you want to do so. And with that, we can conclude this demonstration.
Thank you very much to the Nanoport for that fantastic demonstration. And here, this will just be a summary slide before we transition into some question and answers. But really what I'm trying to highlight with this slide is just the broad range of applications that the Tundra can assist with, and cryo-electron microscopy more generally is able to help with. And so obviously you can do so-called traditional structural determination using cryo-electron microscopy in the Tundra specifically. And we have some examples here that Natalia, during her introduction, elaborated on in more detail. But there's one area that we didn't touch upon, but cryo-electron microscopy can really help with nanoparticle characterization. And so we have some examples here of how that's able to aid research. And we can answer some questions about that in the Q&A if there's any questions there. And really the key point that you would have seen during the demo is just how straightforward and accessible the Tundra is. And so we have a diagram here in the sample optimization line where you're using that structural information that you're gaining from the Tundra to help optimize your sample. And as you saw with the nice resolutions that you can get on the Tundra, you can obviously be satisfied in some cases with that level of data. But if you require to move to a higher resolution, well, you have already optimized your sample. So when you go to a Glacios or a Krios, some other instrument, you then have a lot more confidence that your sample is going to be able to achieve whatever resolution you're looking for. And within all of this, when you saw the Nanoport operating the Tundra, I think probably the key takeaway was just how straightforward and accessible the microscope is. So it's possible that some people approach Cryo-EM with the idea that it's going to be a little bit difficult and maybe not so accessible to a range of different researchers and labs, but really you can see just how straightforward and how much effort has gone into really optimizing and improving the process so that anyone more or less can access cryo-electron microscopy and operate these microscopes. And so I think with that, just once again, thank you to the Nanoport for the great demo, and we'll transition into question and answers. Thank you.
I love seeing that time-lapse video of the installation of the instrument. It was so fun to see how quick and easy it appears to be when you look at it in that method. Also, I love seeing the Nanoport demonstration. Thank you so much, Veer, for that. I've heard from new users who are kind of intimidated in getting started, and really seeing a demo from the Nanoport in that format just makes it much more accessible, as Zuben said, to a wider range of users. So really think this is going to be groundbreaking and exposing more people to Cryo-EM who might want to get started. All right, so thank you to our team who is on for a Q&A session. We do have just a few minutes left for questions, but I'm very happy to start us off with, let's see, the first question looks like a pharmaceutical question. So Zuben, could you please take this one? Does the Tundra have high enough resolution to do single particle analysis for pharmaceutical applications?
Thanks for the question, Tara. And absolutely, the examples that Natalia showed during her talk, I mean, we have resolutions from 2.6 to 4.2 angstrom resolution, and that is very, very sufficient to answer a lot of questions that are relevant to not only academic research but also pharmaceutical research. And critically, as I just mentioned in the little talk 5 seconds ago, the Tundra gives you this rapid feedback on your sample, so you can optimize biochemistry, so you can make really quick decisions. And that's one of the critical factors in pharmaceuticals, is you need something that can give you that quick structural information. And then obviously you can stay with the Tundra, or you know that your sample is going to be sufficient if you move to a higher end collection microscope. So absolutely, thank you for the question.
Yeah, great points. Thank you. And yeah, I think already we're seeing some widespread adoption from pharma companies with the Tundra. So it's nice to see. All right, next question. Maybe Veer or Cedric, if you could briefly answer, how long does it actually take to load a sample and start data collection? So what we saw, was that in live time? What's the actual time?
And, it takes about 10 minutes to load the sample to the microscope, and it would take another 3 minutes to 5 minutes to collect a full atlas, which is your low magnification map of nearly the entire grid. So I would say a total of 15 minutes approximately to get to your first image of the grid. Then if you like the screening part and you want to collect a full data set, it would take about 10 more minutes to do finer alignments, and let's say 5 minutes or so to select a few good grid squares. And so overall, if you just want to screen the grid, it's about 15 minutes of time. And if you want to get started with the SPA data collection, it would be, let's say, another 15 minutes or so.
Perfect. Really amazing that that you could go to loading to imaging that quickly. It is a game changer, I think, in a lot of ways. All right, and maybe just one really quick question for Natalia. Is the sample loading procedure easier than using a side entry TEM?
Oh yeah, that is a very good question. Thank you, Tara. Yes, of course. I have plenty of experience with the side entry holder systems, and they are much more complicated to use. You need to have some expertise to be able to load the samples, especially under cryo conditions, without, breaking the vacuum in the microscope or, contaminating your sample. So with the Tundra, as you can see, it's very, very, very easy of use. I mean, it's very quickly to load the grids, and they are auto grids, so you can totally recover those grids. You have a high connectivity with other systems in our portfolio. So very, 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 it's like going from day to night. So it's a huge difference in improvement and ease of use.
Perfect. Thank you so much. All right. So now we'll wrap up the session. And I'd really like to thank everybody who joined in with us today and who asked these interesting questions. We will make this webcast available along with all of our other recorded webinars. You can find them on our website, or please reach out to any of us here. Our in-person events are continuing around the US and expanding more around the world in the coming months. So if you missed any of these events, please connect with your local contact to schedule something at your own facility. Thank you so much to our speakers and for the Nanoport demo team, and especially to everybody who joined us today. Until the next time, goodbye.